human milk oligosaccharides, which we call HMO, and that's food for gut microbe. That is what people think, but it's little more than what you think. Now, what we just, when you give a breast milk and formula feeds, you can see that when you are giving breastfeeding, that you get a lot of bifidobacteria. Within few days, actually within few days, you can get it, and it remains the same. Whereas the other bacteria like E. coli and Bacteroides, you can see hardly any. But if you give formula feeds to an infant, you can see there is initial rise, but it fades off, and then the predominant E. coli and the Bacteroides takes place in the gut. So therefore, I'm so very nice. Now we are getting the light. Right. Now, so this is just what I wanted to show you. Now, what are oligosaccharides? Oligosaccharides is an oligosaccharide is a saccharide polymer with small number of monosaccharides. So it's a simple sugars grouped together, and maybe about 4 or 5. It's not a long chain like carbohydrates. It's not single sugars, it's multiple sugars, 3, 4, 5 getting together form monosaccharides. Unfortunately, though they are we call them monosaccharides, they are actually glucose and the lactose, glucose, galactose, lactose, and all kinds of sugars. But unfortunately, human beings cannot digest. So oligosaccharides are found only in primate milk. This is the most important part. And non-primate milk has only very insignificant amounts of oligosaccharides. The highest concentration of oligosaccharide is found only in human breast milk. No, even the primates is very little. But in human milk, there is abundance of oligosaccharides. So does this abundance of human milk oligosaccharide contribute towards the superiority of human species in the planet over all other animals? That is the question that we are now raising. When the truth becomes a myth. Now, when we were medical students and even after that, we were taught that the breast milk is sterile. Right? But now there are many studies. This is just one study I'm just quoting. But there are ample studies to show that the breast milk is not sterile at all. It has 10 to the power 8 bacteria per day. The child gets 10 to the power 8 bacteria per day, and there are more than 50 species of bacteria. But fortunately, none of these bacteria are pathogenic. And out of these bacteria, 40% comes from the breast milk, and the rest 60% is from the areola of the nipple. So the mother gives the bacteria to the baby during during lactation. This is a very important point for you to remember. So we were taught that it's okay when the baby is born. We know that when the baby is going through the vagina, and then after that the baby gets bacteria into the gut. So we thought that the bacteria when the baby is born, the gut is sterile. But then they found, which is not so. They found that as soon as the baby is born also, there are bacteria. So then the question comes, if that is the case, so in that case, is the baby in utero, is it in a sterile environment? That is not so, because there are bacteria when the baby is born in the gut. So now we found that there is what you call placental microbiome. There are microbes in the placenta. And so much so, just to quote you, but they found that those placental microbiome consists of only the maternal oral bacteria. It's not the maternal gut bacteria. Placental microbiome is composed of maternal oral microbiome. So that if the mother does not have proper oral hygiene, and if there are pathogenic bacteria like streptococcus mitis and all that in the mother's mouth, then the placental microbiome image will not be normal. So you will not have normal microbiome in the placenta. Right? So now this is just a little bit above oligosaccharides. You can see out of these macro macro substances that you get in the breast milk, macronutrients, you get lactose, you get fat, you get oligosaccharides and proteins. You can see that the human milk has more oligosaccharides than proteins. So it is the third highest component of human breast milk. So much so. And also, so that you can see, as you can see here, you can see your macronutrients and micronutrients. This is water, and out of the macronutrients, out of the macronutrients, you can see the third largest is actually, this is the largest, lipids, lactose, then lipids, and then human oligosaccharides. So this is just a few examples, because now we have isolated more than 212 types of oligosaccharides in the human milk. Now, basically, the human milk oligosaccharide divided into three categories. Actually, two categories. One are called neutral, another one is acidic. Neutral oligosaccharide, there are two types, non-fucosylated and fucosylated. They both are neutral. And then you have what you call silated HMOs, they are acidic in nature. 75% of oligosaccharides that you get in breast milk are actually neutral. That is either non-fucosylated or fucosylated HMOs. And only 25% are silated. Now, this is all these types of things that just to show you that, this is blue, is the blue circular thing is glucose, and this yellow one is galactose, and then this is acetylglucosamine, N-acetylglucosamine, and then fucose is this one, this particular one is fucose, and this one is called actually acetylneuraminidase, that is called sialic acid. So this is called sialate. This, whatever you have this particular molecule, they are called sialated. So they are acidic. But the most important point for you to remember is that the human milk oligosaccharides are also not there continuously. After 6 months, the human milk oligosaccharide disappears from the breast milk. What does it mean? That means that the HMO, human breast milk, has a role to play in the first 6 months of life. Because as you can see, now this is the so-called 2FL, you can see by 4 months, it just become half. And same with this LNT, that is also you can see it goes down rapidly after 6 months. So that means the HMOs has a role to play in the first 6 months of life during the exclusive breastfeeding. So total HMO concentration is higher at early stages of lactation and decreases within first 3 months and rapidly after 6 months. And milk of mothers delivering preterm infants may contain low HMOs, and with the passage of time, the HMO will increase again. So this is different mothers from different continents have different compositions. And same mother has a different composition when you say 1 month, 2 months, 3 months, and so on. And even the same ethnic groups, mothers will have it continuously differ from each other. So there is severe biodiversity of the amount of human milk oligosaccharide from mother to mother. So this is interaction of HMO and microbiome. HMO consumption by probiotics or microbiome and human associated bifidobacteria lactobacilli. These are various studies. This is very interesting because I just put this point. I told you that there are more than 212 types of human milk oligosaccharides isolated in human milk. And they found that certain bacteria will have a preference to a certain group of oligosaccharides. Now, this is just few examples. Now, for example, among bifidobacteria, you can see bifidobacteria infantis strains and bifidobacterium brevis were able to ferment more LNNT. Whereas by bifidobacterium longum and infantis, different strain, they use 3 silylactose, 6 silylactose, 2 fucosylate, 2 FSL, and 3 FL. So you can see different microbes uses different oligosaccharides. So therefore, what does it mean? I told you different mothers have different composition of oligosaccharides. So that means the mother's breast milk determine the microbiome of the baby. Say if say for example, now there are classic, I will tell you. Now this is just to show you that when you are giving human milk, you can see here there is a diversity of microbiota, but they are all abundance of bifidobacteria. Whereas if you are giving control, or so you give something without breast like formula feeds, you can see that it is much less. But if you give a test, that means you give some bacteria, the test bacteria, you can give oligosaccharides. So you can see when you give oligosaccharides, that will have a fairly bit much more than a control group of bifidobacteria. Pathogenic bacteria, you can see, is much less with breastfed babies, but whereas control, it is much higher. And in a test group where you give few oligosaccharides, and you can see there is much less, just like breastfeeding. Similarly, with the potentially pathogenic streptococcus bacteria, you can see almost zero with breast milk. Now, this slide shows very interesting. This is various types of oligosaccharides, but you can see these are all various forms of bacteria, just to show you that the diversity, that different bacteria utilizes different concentration of the microbiome of oligosaccharide, which I have already mentioned. Now, there are 3 ways that the bacteria will utilize these oligosaccharides. One, they can take the bacteria, the oligosaccharides in the just see whether I can get the marker. Sorry, markers I cannot get. But this is, the oligosaccharide will get into the cell, and then inside the cell it is been metabolized. Here it divides into 2-3 particles, and then it is been metabolized. And the metabolism will take place outside, and it will take the sugars inside. The different bacteria will utilize in a different way. Now, this is a very interesting slide. A bit complicated, but in this particular study, what they did was they gave carbon tech galactose to the mother, and then it gets absorbed into the circulation, and then it goes into the liver, and then you get biosynthesis of HMO as well as some of the galactose. So then when the baby is breastfeeding, they found in the breast milk you have lactose, which is carbon tag, C13 tag, and also you have C13 tag oligosaccharides. And when they examined these babies, they found something very interesting. That is, they found certain C13 carbon molecules were there in the in the fecal excretion. So that is after metabolism by the bacteria. That is, of course, we expected. But then they found these carbon tagged oligosaccharides are there in the human cells also and in the plasma. Right? And also they found that certain oligosaccharides will be there in the kidney and also excreted in the urine. So that means that the major role of oligosaccharide is not only to provide food for the microbes, because if it is only to provide food for the microbes, why there is oligosaccharides in the plasma? Why there are oligosaccharides in cells? Why you have oligosaccharides in urine? So that means the people thought that therefore the so-called oligosaccharides are not only the food for microbes, it's having some other functions as well. Now, these are the functions. Now, oligosaccharides, you know what happens if you don't have oligosaccharides, when the pathogenic bacteria gets into the body, if you don't have any, they get adherent into what you call the glycan receptors. This is the glycan receptor. It gets adherent into the glycan receptor and causes infection. This is just like this. So now most of the HMOs actually they have what you call the glycan receptor, like they are they behave like glycan receptors, which is called the decoy receptors. So the pseudoglycan receptor, what happens is all these pathogenic bacteria get adherent to these decoy receptors in these HMOs. And once it is get adherent into this HMO, it will not be metabolized, that will be excreted out of the body. So that means one of the functions of oligosaccharide is to have a decoy effect where they engulf, they get the bacteria get attached to that and will be removed from the body. So that all the pathogenic bacteria will not be allowed to be attached to the human endothelium, gut endothelium. So these are few organisms, E. coli, Helicobacter pylori, and Candida. There are various receptors in those oligosaccharides. This you need not to know. Right. Also, they found that sometimes it modulates the glycan expression, so that it will not allow the bacteria to go and get adherent into that. So not only it has a decoy receptor effect, but at the same time, it can modulate the glycan expression. So the HMO will have dual role in that sense. One is it behaves like a glycan receptor, or it modulates the glycan expression. Also, now they found that it's an immune modulator where the HMOs will provide proper immunity. As you know that if the HMOs are not there, you get what you call Th2 predominance, and when you have Th2 predominance, you get a lot of atopy and allergic diseases and intolerance. But when you have a balance between Th1 and Th2, that you get normal immunity, the so-called real immunity. The immunity does not mean only providing antibodies against something or providing cellular immunity against something. It also tolerance as well as not providing autoimmune disease. They both actually, all these three components are part of the proper immunity. So it's been done by balancing Th1 and Th2. There is another function. Now they found that particularly in newborn babies, when a bacteria gets in and when they produce endotoxins will open up the gates, what you call the gate openers of the endothelium, and then the bacteria gets and all the toxins will get into the body and you get endotoxemia. And now they found that the so-called oligosaccharide will prevent the gate the openness, and therefore it will really make the gut endothelium non-permeable to all these toxins. So In a nutshell, I told you that it behaves like it's a probiotic, gives food for bacteria. That's a major role. And at the same time, it has a decoy effect, decoy receptor effect. And also, as I told you, it has expression of glycan receptors also will be changed. And also, it's balances between TH1 and TH2 response. And also recently they found, because when the I told you that some of the HMOs are there in the circulation in the in plasma as well, so it's there in the blood. And now they found those HMOs really will help for chemoreceptors to work, for leukocyte phagocytosis, enhances phagocytosis. And finally they found that the HMO metabolites of HMO, which are called FFA, free fatty acids, are all neurotransmitters. And they give signals to the brain, which if I have time, I will discuss a little bit. So do we have evidence? Now this is evidence. These are all actually research evidence, not by giving breast milk, but by giving milk which contains those oligosaccharides. The studies had shown that when you give oligosaccharides containing milk, there is, if you give a higher concentration, that you can see that there is a reduction of Campylobacter in diarrhea and also all-cause diarrhea. But in real life, when you are giving breast milk, this is definitely shown that the incidence of gastroenteritis, particularly Campylobacter and all the other bacterial infections, are much less in human milk-fed babies. This is a very interesting field for necrotic enterocolitis. They found that actually certain types of oligosaccharide, the so-called diacylolacto-N tetrose, that is called DSLNT. When the concentrations are higher, that will prevent children getting necrotic enterocolitis. So there are work now going on to use whether we can use this substance as a treatment, because they found that even the breastfed babies, when they get NAC, they found that they have DSLNT much less in concentration. So it is a non-invasive marker to identify whether this child is going to get NAC or not. So and also you can use this as a treatment or prevention of NAC in preterm babies. And also they found the HMOs are facilitating growth of not only the microbes, but the microbes now we know helps to grow a baby in a so-called optimal growth. Helps to make optimal growth to the baby. Silated or acidic milk oligosaccharides promote microbiota dependent growth in models of infant nutrition. This is actually animal experiments. The old neonatal rats, it prevents, as I just told you, that DSA and tetrose will prevent necrotic enterocolitis in animals. So I just see this there is a Malawian study in human infants, and they found that the silated milk sugars promote growth. So growth promotion does not occur with provision of inulin or germ-free mice. So this is a very interesting phenomena. Now we have a lot of oligosaccharides. They are available as prebiotics in the market. I'm sure that you all know there are so many oligosaccharides in the market. And most of them are inulin, or sometimes they have other forms of oligosaccharides. But none of those oligosaccharides cannot be compared with human milk oligosaccharides because they are entirely different. And also now they know that silated oligosaccharides impact liver, muscle, brain, and metabolism, and they may be actually contribute, in the absence of those things, may contribute to metabolic syndrome. So these are all research that to show that 2FL protects against infectious infant diarrhea, 2FL associated with low incidence of Campylobacter and HIV and all kinds of things. This is just to show you that, and then you can see LNNT reduces streptococcal pneumonia load in lungs. So there are so many research going on now, because now we don't know exactly what the role of oligosaccharide, because it's still learning. The more you learn, the more we come to know about oligosaccharides. So why this oligosaccharide? Because the oligosaccharides will provide what you call the proper balanced gut microbiota. So when you have a balanced gut microbiota, as I told you, gut permeability will not be, it will reduce the gut permeability to all these endotoxins and all that. So toxemia and sepsis is much less. And the gut metabolism, growth of the baby, cardiovascular health, the obesity, all this will be protected if you have a normal beneficial bacteria more than the opportunistic bacteria in the gut. But when you have dysbiosis, if you have all these better beneficial commensal bacteria, so the bacteria brought by the breast milk, I told you breast milk every day the mother gives 10 to the power 8 bacteria, and also we can have bacteria coming from the vagina at birth. So all these bacteria will grow in the gut, and by the time the baby, about 4-5 months, the baby's number of cells in the gut is 6 times the bacteria, 6 times the number of cells in our body. You can imagine the amount of bacteria that you have in the body, in the gut. So these are all essentially beneficial bacteria. But when you have dysbiosis, or if you don't have for some reason or other, if you cannot give breast milk, and then there will be dysbiosis, and when you have dysbiosis, there's increased gut permeability so that the toxins can get in, the bacteria will get added into the epithelium, you get endotoxemia, septicemia, and now they found it can cause diarrhea, irritable bowel syndrome, then diabetes, obesity, liver disease, cardiovascular disease, and then brain problems, even Alzheimer's disease, autism. There are a lot of research going on these subjects. So just a little few about HMOs and the brain. There are 2 ways that the HMOs really works in the brain. Some of the HMOs really directly play an important role, like sialic acid in HMOs. Play an important role in brain development, neuronal transmission, and synaptogenesis. And number 2, some of the bacteria produced by, not the bacteria produced by them, but they are food for bacteria, so the bacterial level will go up, and those bacteria also help in synaptogenesis and microglia formation. So HMO provides sialic acid as potentially an essential nutrient for brain development and cognition. And these are research that they have done. 2FL affects cognitive performance and improves learning. 2FL significantly improves the long-term induction and maintenance of the hippocampus, brain involved in memory function. Specific HMOs support behavioral response during stress exposure. These are all experiments. Now, this is the slide that I really wanted to show you. Now, this is a normal mouse with a normal gut microbiome. That's actually what you call specific gut microbiome. And then you can see this is the normal microglia. You have short processes, and then the branching, and they are nicely activated microglia. But if you have a normal mouse, and if you give antibiotics, then you see their microglia is actually different. Their long segments are increased, branches are really very long, the length of processes, the branching is too much. And also if you have a germ-free mouse, the same thing. and the function is also impaired. So you can see when you have a normal gut microbiota, it produces normal microglia in the brain, which produces normal growth. And this may be through the so-called small chain fatty acids. They are metabolites of this. Right. This is actually a very complicated version. I'll have a simple version of this. This is what the people thought of gut microbiota and the peripheral immune system and the central nervous system. So now they know that mainly the impulses are going through the parasympathetic nervous system into the central nervous system. So there's an interaction between the immune system of the body, immune system, and the gut microbiota and the brain. And now we know a lot about this. And for example, there are a lot of, and also not only this, the other diseases as well, like irritable bowel syndrome. And they are now, we know that all are related to gut microbiota. Normal gut microbiota, not like just no gut microbiota, it's a normal gut microbiota. So it is very important as pediatricians for us, at least in the first few months of life, to preserve the normal gut microbiota. I'm telling you, every day I'm seeing patients, small babies, 1 month, 2 months, 3 months, having loose motions, having cough and cold, loads of antibiotics are being given, one after the other. So when a child is having loose motions, which is perfectly normal for breastfeed babies, they give antibiotics that will destroy, knock off the bacteria, and then maybe some bacteria will be remaining. Then you go to another doctor, that GP will know, see that the previous GP has given antibiotic A, so he gives antibiotic B and goes to the third person, he gives antibiotic C, and that will destroy the entire gut microbiota. And the word of caution, as I told you, that the HMOs are there only in the first few months of life. So for a 3-month-old baby, if you give antibiotics unnecessarily and destroys that gut microbiota, and that will not be, you cannot recover that damage. There's no way that you cannot do anything for that damage, because now there are no gut HMOs the way it was in the beginning, and now there is no way to produce that much of bacteria. So for God's sake, the message I want to tell you for all these budding pediatricians, don't give antibiotics in the first 6 months of life unless it is essential, unless there is all evidence to say that this is a bacterial infection. They remember, because I just showed you a few examples. Now they know even the people with autism and people with behavioral disturbances, and now they know they have some degree of gut microbiota dysbiosis. And now in the world, they are doing what we call a fecal microbiome transplant. So unfortunately, you cannot give fecal microbiome to a human being, because nobody will like to swallow somebody else's feces. So all the experimental evidence available to us so far today is for colonic diseases, because you can give fecal microbiome rectally. That's so people will allow. So people who will not have any ulcerative colitis or any irritable bowel, the studies were done mainly with irritable bowel syndrome, and they found that IBS people, when we give a fecal micro transplant, there's 100% recovery. 100% recovery. Because they'll be completely changed. What they do is they give antibiotics and destroy the colonic bacteria, and then they give somebody else's fecal microbiome, which they don't have the disease, and that is probably normal gut microbiome. So there are a lot of studies now, they are on the way in animal experiments, whether we can do for behavioral treatment, people with behavioral disorders and some psychiatric disorders. whether there's a place for FMNT. So a lot of research has been going on. But the important message that I want to tell you is that the gut microbiota is essential for our growth, our CNS functions, our entire growth, and all the metabolic diseases, diabetic, hypertension, ischemic heart disease. They are all actually gut microbiome matters. So it matters because that we provide the perfect gut microbiota because of oligosaccharide, human milk oligosaccharide found in breast milk. Now, this is just to tell you that human microbiota constantly control maturation function of microglia in the CNS, which I showed you. Common cell microbiota affects ischemic stroke outcome by regulating intestinal cells. Acetate mediates the microbiome brain B cell access to promote metabolic syndrome, and there are so many studies. So just function of the gut microbiome, because I'm talking about the normal gut microbiome, which has been really normal for that particular baby, because that is, if the baby is given exclusive breastfeeding for first 6 months, that baby will have the gut microbiome given by the mother. Mother decides what my baby's gut microbiome should be. With this particular bacteria so much, the other bacteria so much, and this one I don't want. So all this composition, that is not only the quality and the quantity of bacteria, will be decided by the mother's breast milk. So in summary, you can see protective function against pathogenic bacteria. I told you what. So immune development, digestive and metabolic functions, and neuronal development. That is function of the gut microbiome. So this is healthy gut. I think I will bypass this. This is just I showed you, because when you are having oligosaccharides, it gives the normal microbiome, and then this human or milk oligosaccharide together with gut microbiome causes actually healthy gut. So it's not leaky. So there are no chance for bacteria to get in. So this is the gut. Now some functions of the gut microbiome. We should talk, cardiovascular disease, lipid composition, periodontitis, behavior and metabolic activities, endocrine metabolism, fat storage, energy metabolism, fat and liver, and also it is not there. So it's behavior and motor activity, and also CNS functions. So the role of gut microbiota, day by day, we are learning so much. And now it is actually, that is why people say that you are talking to the gut and the brain, they are talking to each other. Now, what are the things which affects gut microbiome apart from breastfeeding? Number one is gestation, preterm versus term. Because when the baby is born preterm, there's and very often very little gut or distorted gut, often will do cesarean section. Even otherwise, if you give breastfeeding for a preterm baby, that breast milk doesn't have enough oligosaccharides. And also if you are giving formula, they don't have enough oligosaccharides. So duration of gestation, preterm, that is important. Mode of delivery is also important. Because if it is a vaginal delivery, the first gut, the microbes that you get is from the vagina. But if you are doing cesarean section and then you don't have that, your gut microbes are completely different in a cesarean section baby and the vaginal delivery baby, at least in the first month of life. And after that, when you are going or go on giving breast milk, that it can maybe be normalized to a certain extent. But there's a very interesting study done by in US Baylor College. This particular lady did a very interesting study. What she did was, all the babies for cesarean section, mothers for cesarean section, she divided into two groups. One group, 1 hour before cesarean section, she took a vaginal swab and put it in normal saline. And soon after the baby is born, she put the normal saline swab over the lip of that baby as soon as the baby is born. And the control group, she didn't do it. And when they analyze after 2 weeks, the gut microbiota of that babies who have been given this sip of the, not the sip actually, they touch the lips of the baby with the vaginal swab, had entirely normal gut microbiota, just like a mother who has a vaginal delivery. And when she wrote this article, her conclusion was, you may think that this is probably not a practical thing to do. People will not like your baby to have your mother's feces into the mouth. But she said the day will come that this would be a routine procedure in every labor room. Right? That is the conclusion, because that if you do that, the baby will have a perfect vagina, the gut microbiota, just like a normal natural vaginal delivery. Then mode of feeding, as you know now, formula feeding versus breastfeeding. Environment, this is very important. When a baby is in an NICU, the baby will not be given feeding in the beginning. Then we are giving formula feed sometimes. I talked to you about first 1,000. I don't know whether you listen to that lecture. I gave first 1,000 days one day, and I told you about the environment of NICU, what we should do in the NICU. Then the medication, if we are given antibiotics, and most importantly, proton pump inhibitors. We don't know, we use a lot of proton pump inhibitors in children sometimes. I have seen small kids also they use, because proton pump inhibitors notoriously known to distort the gut microbiota, just like and what antibiotics do. So my last message is that you must never ever try to give antibiotics unless it is really necessary to children, particularly in the first 6 months of life. So if you have normal gut microbiota by giving oligosaccharides in the breast milk, it is what you can see. There's immune tolerance. I told you immune tolerance is very important component of immunity. Immune tolerance is that, you now say one classic example. We have so many bacteria in the gut. I told you that there are 3 to 6 times our gut cells, our body cells. There are bacteria which is 3 to 6 times our total number of body cells in the gut alone. So in other words, that if you take the entire genome of our body, only 10% human and 90% bacterial. So we are not human Homo sapiens. We are Homo bacterians, because 90% of our genome in the body is bacteria. Right? So the importance of this bacteria to be the normal bacteria to be in the gut is that the immune tolerance. So the classic example is that now we have so much of bacteria in the gut, but we never produce antibodies against them. How is that? We never produce antibodies against them. Because the tolerance we have developed, that they are our cells. So not only that, but food tolerance. The studies had shown that the atopy to food is much less when you give human breast milk. And also atopic diseases, for that matter, eczema and others. That is because they develop certain amount of tolerance to food. Right? And then that is tolerance, and also perfect immunity, as I just told you. Then intestinal homeostasis, healthy metabolism, healthy growth, and if we have dysbiosis, there is immune diseases, atopy, asthma, the autoimmune diseases, intestinal diseases, IBS, necrotic colonic cancer, and so on and so forth, diabetes, obesity, cardiovascular diseases, and also behavioral problems, aggressive behavior, or Alzheimer's disease, and so on. And there are so many to mention. I think now time is actually gone. This is actually, I will bypass this. And remember, now we have what you call three types of the substances. One we call prebiotics, that is oligosaccharides. Then you have probiotics, that is the common cell the bacteria that we give orally, which has like bacteria of the common cell. So common cells are not probiotics. Remember, by definition, the common cell, the probiotic, is a bacteria when you give orally, which confer health benefit in adequate quantities. If you give them, it confers health benefit. So that is actually probiotics. So we have prebiotics, that is providing food for bacteria, these good bacteria. And then you have probiotics, that is form of probacteria that you give from outside. And then you have what you call postbiotics. Postbiotics are also as important as probiotics and prebiotics, because postbiotics are actually substance metabolites from metabolism of these oligosaccharides, and those metabolites are neurotransmitters. They have various functions. They keep the stool loose, and there are many other functions in the gut integrity, maintaining the gut integrity. So you have three types: prebiotics, probiotics, and postbiotics. In conclusion, HMO is the natural prebiotic made to colonize a distinct gut microbiota for her infant. The word her infant. So it's a distinct gut microbiota for her infant. It enhances both innate and adaptive immunity. So it has decoy receptor effect, inhibits epithelial adhesion, balancing Th1 and Th2 immunity, restore leaky gut in gram-negative sepsis. Modulation of leukocyte rolling and adhesion, I just told you. Prevention of non-communicable diseases, type 1 diabetes, IBD, strokes, and CNA diseases. Postbiotic byproducts to communicate with brain, and establishment of symbiotic gut microbiota for lifelong health. So this is the gut-brain axis. This is how they talk to each other. And then this is, if it is dysbiotic, you can see it is utter chaos. Now this is actually the slide I want to show you, my last slide. So the head brain is talking to the gut brain and say, hello, gut brain. The gut brain is talking to the head brain, say, hello, head brain. So the head brain says, oh, Gutty, what is the matter? Then he said, tell this man to stop this talk. Let's go and have our dinner. Thank you very much.